It's my pleasure to talk to you about precision medicine and clinical trials. Essentially, things that you hear about on TV about what's new and what's changing in the field of cancer. I'd like to try and not overwhelm you with lots of details and just walk you through things that I've experienced myself and use examples of patients, if you will, to describe what happens. But the talk is about transforming the treatment of cancer from kind of a shotgun approach where we were treating cancer cells which were rapidly dividing with a cocktail of chemotherapy drugs designed to essentially wipe out or destroy proliferating or rapidly dividing cells. So we treated all cancers the same with varying cocktails of drugs, chemotherapy drugs. So in addition to destroying dividing cancer cells, there were lots of other parts of our systems that were hurt in collateral damage, if you will. So lots of side effects and troubles, as you've seen in pictures and so on, of bald people getting chemotherapy with mouth sores and protected in highly protected environments sometimes to prevent infections and so on. So it was quite challenging. The era of knowledge gained at the bench from laboratory scientists that identified amazing molecular details in cells allowed then the ability to potentially design highly specific ways of treating cancer, and therefore this concept of precision medicine and highly targeted therapy. Clinical trials refers to experiments involving human subjects. So we are taking people with cancers and testing new drugs in clinical studies, or attempting to, and hence the term clinical trials. In order to understand precision medicine and clinical trials, it's best to use an example of a specific disease where this became kind of the prototype for precision medicine, clinical trials, and at least initially was hailed as the one example of which we were almost near the end of cancer as a disease. But as you can see, cancer is a wily beast, and there are so many different ways by which it can escape our ability to control it. But I want to start with the first example of a treatment for a disease called chronic myelogenous leukemia, or CML. Chronic myelogenous leukemia is a leukemia that mainly affects adults. It's a disorder of the blood, a cancer of the blood, if you will. And it took about 160 years from the time the disease was first described to finding an effective precision medicine targeted treatment, if you will. And it's worth spending the first few minutes of this talk just understanding this, so that it'll give you a background of what it takes to move from a mere description of a problem to finding treatment. So the first description of chronic myelogenous leukemia as recorded in at least the Western literature is in 1800s. Virchow in Germany and John Bennett described patients who had peculiar changes in their blood. Very high levels of white blood cells of a variety of types were identified. And the description was associated with people who had enlargement of organs like the spleen, which in all of us is really about the size of your fist and sits under the left side of your rib cage. In patients with chronic myelogenous leukemia, as the leukemia gets worse over time, this fist-sized organ balloons to fill the entire belly, the whole abdomen. And patients have lots of troubles from pain, discomfort, and this dreadful blood disease, which killed all patients with that diagnosis. And this was even before we could have special stains to look at what a white blood cell was, a red blood cell was, and so on. So this is a fairly early descriptions. And it took a long time before a molecular defect was identified in patients with this leukemia. I started my career as a hematologist in 1985. So, in a very short period of time, I have, been the beneficiary of taking a dreadfully fatal illness. And when I was a fellow in training at Boston City Hospital, learning to be a blood and cancer doctor, if you will, patients were still being treated with a cocktail of chemotherapy drugs. Some of these were pills called hydroxyurea. Some of these were injections called alpha interferon, and people were miserable. Virtually every patient of mine with chronic myelogenous leukemia at this time progressed on, failed treatment, and those who were lucky enough to get bone marrow transplants, which was applicable only to a very small number of those patients, seemed to do well, but the rest of them all died. The treatments were associated with horrific side effects. Patients at the time of diagnosis, and by now we could detect this leukemia fairly simply by doing a simple blood test, actually were pretty fit. Were not very sick at all. But I ended up making them horribly ill with the treatments for this disease. So alpha interferon was used in high doses, and it transformed people who were feeling very well into literally shells of themselves. It had global effects. It made people weak, fatigued, like they were having a bad case of the flu every single day. And it was terrible to treat this disease. But later, this drug called Imatinib, a single pill, just transformed our ability to treat this. And it was effective in 98% plus patients with this disease, just taking it once a day. It was extremely well tolerated, and people didn't seem to have many serious side effects at all. And almost all of them appeared to be cured, if you will. That is to say, this molecular defect, which could be identified, completely disappeared. They had complete molecular remissions. So here I am as a trainee hematologist, budding hematologist at Boston City Hospital in 1985. And in this period of time, where I then went into practice as a young faculty person, so specific agent, Imatinib, became approved for the treatment. And from 2001 to just a few months ago, for the first time, I actually stopped treatment on a patient who was in complete molecular remission. So now we're no longer worried about curing the disease. We're trying to see whether some patients in whom the disease disappears seemingly in molecular remission for decades, can we actually stop the treatment, and will the leukemia stay gone? That's where we are now with this particular disease. So this disease really excited the world of cancer, both in terms of scientists in the laboratory as well as those of us who took care of people at the bedside. Meaning, if we could find a single drug that would hit a specific mutation, cure the disease with minimal side effects, we're done. In order for that to really be true, then we have to understand things a bit further. So in order for us to really have Imatinib, which is a pill that specifically targets a molecular event and cures disease, there was so much information that needed to happen in the background to allow us to really reach this spot. And we can start with chromosome banding. So people in the 1960s looking at chromosomes, human chromosomes, identified that in patients with chronic myelogenous leukemia, there was a funny looking chromosome. So Noel and Hungerford in 1960 said, oops, there's a funny looking chromosome in every patient with this disease. And it was in the 1970s when Janet Rowley identified that this funny looking chromosome was a flipped translocation between chromosomes 9 and chromosome 22. And it was much later that they identified that there was this molecular defect where an oncogene moves from chromosome 9 to chromosome 22, and that was ABL. And it was in the 1990s when investigators in Boston, David Baltimore's lab, took this molecular fusion product, which was seen in every single patient nearly with chronic myelogenous leukemia, put this transposed oncogene, if you will, into animals, and they developed chronic myelogenous leukemia. If they didn't have this, they didn't develop the leukemia. So this was shown to be the event that drove a normal system to transform into a malignant system. In order for folks to know a lot about oncogenes then, in 1911, Peyton Rose had shown that using a funny filtrate from a tumor, like essentially crushed a bunch of sarcoma cells, filtered it through a filter paper, injected it into a rooster, and the rooster developed a sarcoma. And this was in 1911. And this was later identified to be an oncogene called SARC. This soon exploded the field of tumor virology, and these were shown to be part of normal cellular DNA, having specific roles in cell proliferation, and that if these pieces of DNA became abnormal, they were oncogenes and drivers of malignancies. This whole field of protein phosphorylation then in the 1930s led to identification of very unique phosphorylation of proteins. Phosphate groups that were moved to proteins enabled these proteins to then drive signals within cells. So this identification of tyrosine kinases as signaling proteins, if you will, was a very important step. And so all of this had to happen before folks realized, look, if chronic myelogenous leukemia had a single gene mutation that was driving the disease, and we knew that this fusion protein was a tyrosine kinase, can we not inhibit this phosphorylation of this abnormal tyrosine kinase? So scientists at Novartis Pharmaceuticals partnered with a physician called Brian Drucker, and they began to screen whole libraries of agents that they had in their pipeline to see if they would block BCR-ABL tyrosine kinase. And tyrosine kinases essentially bind to ATP, adenosine triphosphate, and they transfer phosphates to tyrosine residues. And as soon as the tyrosine is phosphorylated, it seems there is cellular proliferation. So if you blocked this binding site between ATP and this tyrosine kinase, maybe things would stop, cells would stop proliferating. And that's exactly what happened to one specific tyrosine kinase inhibitor in their library, and at that time it was called STI-571. This particular molecule seemed to have dramatic cellular inhibition of BCR-ABL. And so they gave it in a phase 1 trial to 53 consecutive patients who had failed all standard treatments for chronic myelogenous leukemia. I believe it was 54 patients or so, but 53 out of 54 patients, when they reached a dose of 300 milligrams of STI-571, every one of them were in hematological remission. Remember, they had failed every known drug and treatment for CML. All of them went into a remission. So they went to a phase 2 to study what would be the importance of using this in patients with newly diagnosed chronic myelogenous leukemia. Will it work? And indeed, again, 95% of patients exposed to this in a phase 2 study responded with a complete response that happened very quickly. Within 2 to 3 months of starting this pill once a day, complete remission was seen. So they went to a phase 3 where they had to compare this new drug called by then Imatinib to the best standard of care, which was interferon and hydroxyurea. So multiple centers in the US and in Europe combined forces. So in a 7-month period, well over 1,000 patients with chronic myelogenous leukemia were enrolled on a study where they were randomly assigned, so almost like a flip of a coin. Half of them were treated with 400 milligrams once daily of Imatinib, and the rest of them randomly were treated with the best available doses of alpha interferon plus hydroxyurea, and that was the standard of care. So well over 1,000 patients randomly assigned, roughly 500 in each group, and the results were quite striking. Within 3 months, 90+% of patients on the Imatinib arm were in complete hematological response versus less than about 50% of patients with the standard of care. Most patients on Imatinib achieved a major cytogenetic or complete cytogenetic response, whereas only 20% of folks with standard of care achieved that kind of response. And all this really rapidly changed the way we took care of this disease. And so it raised an excitement. So if we can do this with chronic myelogenous leukemia, can we do this with breast cancer, lung cancer, stomach cancer, and so on? And it was pretty exciting that this held a glimmer of hope. So this concept of translational cancer research then is extremely important. As you have seen in this example, an observation that is made either at the bedside or in the laboratory then is studied in context of a body of medical knowledge. Then there's an hypothesis in the previous example, does BCR-ABL really drive the tumor? Somebody showed it does, and so could we block that? Then they make that discovery at the bench in the pharmaceutical company in that instance, and they move the drug into clinic. We see how effective it is, and then we make further observations. How many patients does it work in? And if it doesn't work, why doesn't it work? And they take it back to the lab and figure out in those that it didn't work, do they have a different mutation and so on. So this cycle of translational cancer research is an extraordinary enterprise and requires large-scale collaboration, as you will soon see in the rest of the slides, with different examples of more common diseases than chronic myelogenous leukemia. So let's change the subject from an uncommon leukemia like chronic myelogenous leukemia to a dreadfully common condition, lung cancer. It's the #1 killer of men and women across the world. More people die from lung cancer than many of the other common cancers combined. And there are 2 major types of lung cancer, if you will. Lung cancer that is consists of small cells, which is an uncommon type of lung cancer, and therefore it's called small cell lung cancer. And all the rest, for lack of a better term, 85% of lung cancer is non-small cell lung cancer, of which there are 2 main types, adenocarcinoma and squamous cell carcinoma. I won't bore you with the rest of the details, but this is of what it would look like when you biopsied the lung tumor in a patient who presents with, say, cough, phlegm, chest pain, and an X-ray shows a big lump in the lung. We put a tube in, or we put a needle into that area, take a biopsy and stain it. This is what adenocarcinoma looks like under a routine hematoxylin eosin stain. And we used to simply use this as a single entity, adenocarcinoma of the lung, and we threw a bunch of chemotherapy drugs at this. Some people responded, most didn't, and for most of my career, lung cancer has been a terrible disease to treat because most of the time we diagnosed this in the advanced stages and we had very little to move the needle. In fact, I used to talk to my mentors and who spent all of their time with nothing but lung cancer, and they didn't give up. They just wouldn't give up with the fact that for 20, 30 years, all we had was a cocktail of chemotherapy drugs that only helped a small fraction of patients, until folks in the lab were looking at molecular tools to study adenocarcinoma. Why do some do well, others don't? And this panel here is a technique called break-apart FISH, or in situ hybridization. So there's the green signal, if you will, stains the 5' end of a oncogene called ALK, or anaplastic lymphoma kinase. The red stains the 3' end. If they're joined together, as in this area, it's a wild type or normally occurring ALK gene. But if they're spread apart, that suggests a translocation. And this was first described in patients with an uncommon illness called lymphoma. So it was shocking to find that adenocarcinomas of the lung, a much more common beast, a small fraction of adenocarcinomas had an ALK translocation. Now this resulted in activation of a tyrosine kinase. So the ALK protein, the abnormal ALK protein, could be stained by immunohistochemistry. And in the adenocarcinoma portion, they stained brownish, whereas the blue is normal lung epithelium. So it stained only the adenocarcinoma cells. This is a representation of an assay called an RT-PCR that shows a junction between the ALK and its partner protein called EML4. But this then showed that in adenocarcinomas, the ALK translocation would then activate a tyrosine kinase, for which then we had the possibility of using a drug that could target this. And would it work? Again, an observation of an ALK translocation in adenocarcinoma of the lung resulted in background knowledge already that we know that tyrosine kinase inhibition works. And can we diagnose, can we find one that would work in this particular ALK translocation? This is one of my patients last year, presented with abdominal pain. And when we examined him, this is a CAT scan showing his liver. It's about 8 times the normal size. The liver was filling most of the abdomen, and he would just hold his belly when he walked, when he came into my clinic. And initially, it was so frightening at the bedside that the thought was, and in San Antonio this is an epidemic, that this was a primary cancer of the liver. We were so focused on the fact that the liver was huge, full of cancer. This was biopsied, shown to be an adenocarcinoma, and it could be a primary liver cancer. Except when we sent this tissue for profiling, and I'm sorry that it doesn't project well, by immunohistochemistry, they identified the ALK, abnormal ALK protein. We didn't have enough tissue there to do what is called next generation sequencing, but at least by immunohistochemistry, it identified that this had an ALK translocation. So we did appropriate screening to see if there was a lung tumor, and indeed there was a smaller tumor in the lung. By now we had a drug that could hit ALK, and this is the same scan that I showed you at baseline, which is in May 6th of 2017. This is the liver CT with all of these blotchy areas full of tumor. And this is July 11th, 2017, after he had been on a drug called Crizotinib, again a pill that he takes daily. And you can see dramatic shrinkage of the areas of the tumor. The liver regressed quite significantly, and he had no symptoms whatsoever, no side effects from the drug at all. So this would be a happy ending if I told you that, like the patients with CML, I'm talking about taking him off the Crizotinib and everything is happy. But that's not the case. And again, we go back to this issue of translational research. People who go into remissions with lung cancer and Crizotinib and ALK become resistant to Crizotinib. And there are so many different mechanisms by which these cancer cells can escape the control of Crizotinib. And so we have lots of work to do. So although there was one drug, Crizotinib, when we first identified the importance of ALK in adenocarcinoma of the lung, I have a lady in clinic now who's on her 4th ALK inhibitor, a drug called Brigatinib. So she has failed 3 different ALK inhibitors and is still responding to a 4th variant that is a much more potent inhibitor of the tyrosine kinase. And this is a lady who a year and a half ago presented with lung cancer, adenocarcinoma, with an ALK translocation, with brain metastasis, bone metastasis, and liver metastasis. And if you looked at her in my clinic, she looks like she stepped out of Vogue. She's always like impeccably dressed, and you would never tell that she has widespread lung cancer. And she's fearless and in my face and will not let me stop finding the next. She knows she's going to relapse, but she's already on my tail. What are we going to do next? I mean, this is how inspiring it is to see this whole process evolve. So this is what happens in lung cancer now. I'll be very brief about that. So this is non-small cell lung cancer. I told you that we have two varieties. One is called squamous cell, the other is adenocarcinoma. Adenocarcinoma, you can identify mutations. EGFR stands for epidermal growth factor receptor. So we've identified activating mutations in the EGFR receptor, for which we have tyrosine kinase inhibitors, oral agents that produce dramatic responses, including in the brain. I talked about ALK translocation, and then next generation sequencing producing so much information, some of which we have drugs to match, and others we have no clue what to do with. Squamous cell cancers, unlikely to have any of these mutations, but they might be amenable to immunotherapy. There are new aberrations of the immune checkpoint pathways for which we have specific drugs. And so you see the ads on TV for Keytruda and Opdivo, drugs that are exploiting the immune checkpoint system to control lung cancer. So the diagnostic algorithm for lung cancer now no longer stays with just these simple histopathological squamous cell adenocarcinomas. Adenocarcinomas are being split apart into a whole range of diseases, really, for which there are dramatically different treatment approaches, and the same may happen for squamous cell cancers as well. So look at the timeline between identifications of these mutations and the availability of treatment. It's quite exciting. Unlike the 160 years between description of CML and Imatinib, you have EGFR mutations identified. Gefitinib is used to hit that activating mutation. Here's the ALK timeline. You have EML4 ALK detected in 2007, and by 2011, people are on Crizotinib doing well, and now there are multiple agents as well. So it's getting shorter and shorter and much more complex in lung cancer. So very common cancer, extraordinary timeline in terms of understanding background disorders and new treatments. We talked about the EGFR, we talked about the ALK, very short timelines from immune checkpoint identification. In fact, 3 years between the identification that you could inhibit PD-1 or PD-L1, which are problems in the immune checkpoint pathway, to full approval of an agent. It's a very short timeline. It might seem long to you when I say 3 years, but I'll show you in the next slide what happens. How do we actually take a drug that is known to be effective in an animal model and then use it for the first time in human beings, and then show that it's the best way to treat that cancer? How quickly, how do we do this? And that's by clinical trials and trial designs, and I'll just take you through this fairly simply. So there are, in the modern era of molecular diagnostics, specific ways by which trial designs have emerged. Let's say in this instance, we call it a basket clinical trial. We have a purple pill, let's just say it's Crizotinib, which is an inhibitor of ALK. We could try that in any patient who has, say, a lung cancer, a liver cancer, a pancreatic cancer, a colon cancer. If you identify any ALK in these cancers, you could potentially try treatment with an ALK inhibitor. So this is called a basket trial. An umbrella trial, on the other hand, screens literally any patient with any cancer and tries to identify a genetic mutation matches that gene mutation to a specific drug. So everyone would say this particular gene mutation is linked to a drug that works only for that mutation. Similarly, people with a different mutation, you choose another drug that is known to be active in that mutation, and so on. So under a single trial, there will be cohorts, several different cohorts being tested, matching the genetic aberration with a specific drug known to play a role in inhibiting that pathway. So those are the types of trials that are being conducted these days. Traditionally, though, we talked about phase 1, phase 2, and phase 3. Phase 1 is simply trying to find whether a drug is safe in humans. Once we know that it's safe and we find a reasonably safe dose, then we move to phase 2 to find out if it is actually active in a group of patients with that disease. Once we know that it's active, then we randomize patients to the new drug versus the best standard of care, often requiring, look at the sample size, maybe 20 to 30 patients in phase 1, 550 to 100 patients in phase 2, and sometimes thousands of patients in phase 3. We're lucky, like in chronic myelogenous leukemia, if we could complete a 1,000 subject study in 7 months by having large-scale collaboration between Europe and the United States. It's very, very challenging to do when it's an uncommon disease, if you will. So the newer ways of shortened time to approval takes genetic mutations and enriches cohorts. So if you have patients with lung with an ALK mutation, colon with an ALK mutation, you keep adding patients to these different cohorts and expanding the numbers if you see an effect, so that drugs can be approved faster and faster. And this is happening, shortening the time, but leading to all sorts of other questions that come up as well. So let's sort of end this talk, if you will, by describing one such umbrella trial called the NCI MATCH clinical trial. This is a precision medicine trial that explores treating patients based on, exactly what I've shown you examples of. Taking patients who have cancer, identifying a genetic mutation, linking that with a specific known drug that could affect that pathway, and seeing if we have responses. So at the outset, they wanted several thousand people to have biopsies for which we could screen for particular genetic mutations, see if we had a drug that they could match, offer it to the patient. If the patient said yes, they are enrolled on the study. So not everyone will have an abnormality, only some fraction of those will, and patients who have that, you inform them and you see if they will enroll on the study. Thus far, it appears that about 6,000 samples have been screened, of which there is an about 18% match rate. It's not that high. So it's not like CML, where every single patient has that specific mutation. Sometimes in all sorts of cancers, we're talking about all different types of cancers, you may not have a specific match. But once you identify the match, most people seem to enroll. At least 70% of folks enroll on the study. So the enrollment to this has actually been complete. So in less than 2 years, about 6,000 patients have had their tumors sequenced, mutations identified, trials identified. And this, just to give you an idea, requires dramatic collaboration between institutions to have that trial open at their institutions, to have the wherewithal to talk to patients, ask permission to have their biopsies sent for sequencing, get the details, come back, talk to them, get their consent, etc. Requires a lot of effort. It requires a lot of laboratory effort. It's one thing for me to talk to patients in clinic. How are these 6,000 samples being sequenced? Well, Mass General, MD Anderson, Yale. There are lots of institutions that have partnered to sequence these tumors so that we can do it faster and faster. So again, clinical collaboration, laboratory collaboration. These are the treatment pathways for which we are looking at mutations for which we may have drugs. And it covers a variety of mutations affecting lots of cellular domains in the MAP kinase pathway, the PI3 kinase pathway, DNA repair pathways, and so on. So how does this work? So these days, in patients who are failing standard treatment, we ask patients permission. There are commercially available laboratories that can do limited sequencing of tumors and give us at least a hint that there might be an abnormality. So we do this almost routinely in advanced cancer patients these days. The lab runs a standard test and sends the result, and it might flag this on the report to say that there's a specific gene abnormality that might qualify them for one of the dozens of arms on the NCI match study. And then we decide to talk to the patient and enroll them on this. And so here are just the next few slides showing you a bewildering array of mutations. Here's the prevalence rate of these. So it requires, lots and lots of patients to be screened to see how many of them would have that. We have drugs that can match these different mutations. Many of these cohorts have been completed or closed from already accruing enough patients with these mutations, and results are awaited, and I'll share some with you. As we learn more and more of new mutations for which there are drugs available, we expand cohorts like I was talking to you about. And it opened with 10 arms, very shortly 24 different arms, expanded this year to about 40 different arms. So the logistics to keep all this straight is quite extraordinary. And here are some results that were presented at the American Society of Clinical Oncology last year. Arm 1, the PI3 kinase mutation, they used the drug called Tacelicib in 65 patients. It didn't work. No responses were seen. In other words, we measure responses by whether we can see the tumor shrink. They didn't see any responses. They looked at a subgroup of patients who had stable disease. It didn't get worse, but it certainly didn't get better. Similarly, HER2 oncogene positive tumors with a drug called TDM1, there were some responses in 3 of 37 patients. So these are not all breast cancer patients. Could be any cancer that has a HER2 abnormality. Similarly, FGFR mutations using this drug, 50 patients, a partial response seen in 4. This raises a whole other translational research questions. Who are these lucky 3 and lucky 4? Why did it work? Why didn't it work in the others? And there's a spin-off of trying to understand that as well. And these are protocols that are closed to accrual as of June 25th of this year, and results will be forthcoming, and we will see if there are hits or misses as we go along. I want to stop by, again, first of all, thanking you for your attention for what is sometimes a very challenging area to get across. I struggle with it all the time. But I think one take-home message is that cancer clinical trials are really, really important. And I admire my pediatric colleagues. Most children in this country are enrolled on clinical trials, always asking a question, how can we get better? We'd like the same thing to happen with the much more common problem in adults. I talked to you about translational research. It is so important for me to partner with my colleagues at the bench and beyond if we are to really move this field forward. And I am so inspired to tell you that at this cancer center there is large-scale collaboration, open doors, constant communication to make this happen. The clinical trial staff at the Mays Cancer Center are amazing. They help me talk to people, the informed consent issues, keeping me in line with making sure that I do whatever is needed and demanded of the study that may not be standard of care, that I'm reminded to do this in the right sequence so that we don't mess up the study. The nurses and staff who help us take care of people who are on these clinical trials, and most importantly, patients and their family for participating in studies like this. Thanks very much. I'll take your question.